Thank you. Okay. Thank you very much. Okay, it's me now. Finally. So, ladies and gentlemen, members of the Royal Dublin Society, family, friends, president, members of the press, mum, welcome and thank you for coming along this evening. So before I get into the actual talk itself, I've heard some very nice things said there. In many ways, it's a little bit like being at your own funeral, hearing some of that stuff that was very gracious of our speakers to say. So I'm going to talk about cell death as it happens, but at a micro level. Now, all of us here have lots of cells, obviously. In fact, all of us are literally teeming masses of cells all cooperating together. And I'm going to talk about that subject, something that actually we take for granted all of the time. Clearly, most of us know that we're made up of cells, but it's only in recent years that we have begun to really appreciate the complexity of the cell. And the cell is often depicted as a relatively simple round bag of molecules. And in fact, as more and more powerful microscopes and analytical techniques have become available to us, we increasingly are able to look inside the cell and appreciate its wonder and complexity. So, before I start on the talk proper, I would like to say, of course, I'm very honoured to be the 39th recipient of the Boyle Medal, and I would very much like to thank the Royal Dublin Society and the Irish Times for their support of science in general, and obviously for their support of the Boyle Medal. So the cell is the basic unit of life. I think most of us learned about the cell at school, and as I say, we don't think about it a great deal. Clearly, progress is being made all the time. But as we increasingly resolve and peer into the cell, we realize with each passing year that cells are really amazingly complex. And sometimes we hear things about nanomachines being developed that will make us immortal in the next 20 years. I've heard that said a couple of times on radio programs over the last couple of years. And I always laugh to myself when I hear those kind of predictions, because we're still literally 20 to 30 years away, even from understanding how a cell works. Never mind designing nanobots to go in there and make us immortal. So as I mentioned a moment ago, we are comprised of literally millions and millions of cells. In fact, we have 10 million cells, 10 trillion cells in the average human body, all working pretty much in harmony to keep us alive, to defend us from infectious agents. And obviously, we don't have to consciously think about that. But obviously, these cells also there's an uneasy truce going on, because these cells have to rely on one another, and they have to make sure that everybody is doing what they're supposed to be doing. And as I'm going to tell you, for cells that don't do what they should do within the cellular society, I'm afraid capital punishment is frequently the major outcome. So in most cases, if a cell actually behaves abnormally or is defective or infected in some way, what actually happens is that the cell actually commits suicide. Now, most of the time that happens spontaneously. There's an actual dedicated machinery to kill cells within every cell, and it's very complex, and that's what I've been spending my last 20 years or thereabouts investigating. And that's a remarkable fact, that most of the time when cells die in our bodies, they actually die in this programmed, self-regulated manner. This machinery is switched on in those cells, and it actually causes the cell to auto-destruct. Now, there are good reasons for that, which I will get into. And because this process, as I say, helps to make sure that in the cellular society, everybody is cooperating and doing what they should do. Because that's the basic understanding of organisms like ourselves that are made up of millions of cells, that if you divide the labor, you make sure that everybody does what he's supposed to do. And if they don't, then you get rid of them. So, as I've mentioned already now, cells are really amazingly complex. They're also very beautiful. And using labeling techniques that enable us to label specific molecules in the cell called proteins, and they're really the molecules that do most of the work within the cell. And I'll tell you later on that cells are actually comprised of about 10,000 different proteins, but with up to 500,000 copies of each protein. So even in a single cell, you have literally tens of millions of individual protein molecules all working together cooperatively to keep that cell working efficiently. So cells are complex. So although they look deceptively simple, so each of those, unfortunately, my pointer doesn't work on the screen, but there are 4 cells there. They look relatively simple, but as we develop methods of increasing power of resolution, we realize that there are many layers of complexity within the cell, lots of different compartments, and many, many different proteins all cooperating together in a way much like hundreds of bees cooperate within an individual beehive. You have many, many beehives within an individual cell with literally hundreds, if not thousands, of proteins all cooperating in their own particular hive, doing the particular job that subset of proteins do. So as I've mentioned already, the body is essentially a big box with literally 10 trillion cells, 10 million cells, 10 trillion cells inside. But it's not static. You're actually making cells right now as we speak. In fact, over the next 30 minutes, you will actually make about 10 million cells, and you will kill about 10 million cells in your body. Hopefully, I won't accelerate that process of cell death. Maybe in your brains a little bit, but this is quite natural. Happens all the time. Most of us are familiar with the concept that cells divide. And but the only reason that cells divide when we're adults is because cells are dying all the time. Because we don't grow throughout adulthood. Once we reach the age of 18, give or take a few pounds, we essentially don't actually grow physically anymore once we reach the age of adolescence. So the main reason that cells divide is because there's such a rate of cell death going on. And as I say, literally tens of millions of cells are dying in your body every hour, and they're being replaced by the birth of new cells that are there essentially to replace them. So as I mentioned at the outset, it's essential that all of these millions of cells do the right job, and there are a number of rules that are in place. Simple and crude rules that cells are not allowed to violate. And I'm going to stress these quite simply, but a basic rule is stay where you are. It's a bit like if you own a house, you don't go into your neighbor's house. Okay, that's a certain rule in human society. You stay in your own house. And cells also have that rule. Cancers break that rule. They metastasize into other tissues. But most of the time, that doesn't happen. If cells move where they shouldn't move, they're killed. You don't eat your neighbor. That's a fundamental rule, I think, in human society. And it's also a pretty important rule in cellular society. In our bodies, cells are not allowed to eat each other except on special circumstances. In fact, during this process of programmed cell death, as I'm going to tell you later. You don't build an extension. You don't double the size of your house in human society without permission. If you don't get planning permission, then the council or corporation will come and knock down your house. In the same way, in our body, cells actually need permission to divide, and that permission is given in terms of special cell-cell communication molecules called growth factors. And as I said earlier, cells have to do their job, and they're not allowed to harbor bugs, aliens that could potentially take over the whole cellular society. So if a cell is infected with a virus, it's meant to do the honorable thing and kill itself. And also cells are not allowed to act weird. If they act or misbehave in some strange way, they're also violating the rule. In all of these cases, the primary punishment, as I said, is death. So there are rules in the cellular society. Death is the penalty. There's no warning, no fines, no prison. In most of these cases, if cells actually transgress the rules, then they die. But they die in a special way, a way that we've been investigating. This death is called programmed cell death, as I've mentioned, and it turns out it's pretty complicated how it's actually regulated. So there's a constant flux, a throughput in our tissues, like a conveyor belt, with cells being born on one side and going through and aging and becoming dysregulated or infected or injured in some way, and then they die through this programmed manner, and that's all very predictable, and as a result our tissues are constantly replenished. As I mentioned, as you're sitting here, there's cells dividing and cells dying. So in fact, we make many more cells than 10 trillion during our lifetimes. We make 1,000 times that. So when sometimes people say they're not the same person they were last year, that is literally true in most cases. There are some exceptions to that. Some cells live for a very long time, like cells in our brains, and some cells live for a very short time. Some cells in our immune system, for example, only live for an average for 12 to 14 hours before they're replaced, and they are replaced therefore at a tremendous rate. So there's this delicate balance between cell death and cell division that goes on in our bodies, and if that balance is shifted, so if you have too much cell division, not enough cell death, you can get a cancer, and if there's too much cell death, not enough cell division, you can get a neurodegenerative disease such as Alzheimer's or Parkinson's disease. So from a point of view of health and disease, in most major diseases, in fact, the controls on cell death are disrupted in some way. So how do cells die? These are I'm going to show some pictures of cells at various junctures during the talk. These are mainly taken in my lab at Trinity College using a fairly high resolution type of microscopy, and this enables us to examine to a fairly high degree of detail events that take place in the cell. So do cells die by accident or design? Well, it turns out that cells die by design, and that's the program mode of cell death that we've been investigating, but they also die accidentally. And I'm going to use analogies for those two types of cell death in a moment. But it wasn't always appreciated that cells could die by design. In fact, for many years, this is how cell death was considered by the scientific community until really the late 80s. It was considered cells are going along, minding their own business, and then one day something terrible happens. And in fact, when I started to work on cell death, that really was the prevailing view that held amongst about 99% of scientists, that why would you study cell death? It's not a very interesting process, nothing much happens, we can't really change the outcome, so why bother study it? That's kind of the car crash view of cell death. It's random, it's chaotic, and it's not really regulated or controlled in any way. Well, in fact, we know, as I've said already, that in fact, most of the cell death that takes place in the body is of this variety, of this programmed cell suicide. So there's a dedicated machinery that's within every cell in your body that can be switched on either by factors externally to the cell or by the cell itself. And the machinery is what we've been trying to understand over the last 20 years. Now, in extreme cases, when we suffer extreme injuries, such as extreme trauma, burns, compression injuries, certain toxins and so on, if we drink large amounts of alcohol, we can get this other type of cell death called necrosis, and most of us have heard of necrotic lesions and so on. And that was the major mode of cell death that was recognized really until the late 80s, early 90s. So superficially, in both cases, cells die, but the way they get there and the consequence is very different. Now, I sometimes like to show an analogy when people ask me, well, why does it matter? That this person isn't dead. Actually, he's meditating, and this is apparently a pretty advanced form of yogic meditation. And this person isn't meditating, but so it's ended up in a superficially very similar predicament, but has got there, using a very different upstream stimuli, and the consequences are very different as well. So let me get into how I got into cell death. These were the bad hair years. This is me on the left-hand side there. And this is when I started my PhD in UM Maynooth. I began working with Professor Tom Cotter, as you heard earlier on. And at that time, actually, Tom was working on leukemia, which is a type of blood cancer. And I was given a project to try to understand how we could enforce the maturation of these leukemic cells, because one of the characteristics of leukemia is that cells get stuck at an immature stage. In this particular leukemia, they get stuck at this very early promyelocyte stage. And what we were trying to do was enforce them to mature to the normal, desirable cell, the neutrophil, And so I was doing this, and also I was treating these cells with the chemotherapeutic drugs that were used to treatment leukemia at the time. And I consistently noticed these rather strange looking cells. Normally, a cell has a nice round blue nucleus, and at least in this, when viewed in this way. And these cells look like a bunch of grapes, essentially, and their nucleus had been shattered into many, many pieces. And they looked very odd. And I was seeing them for quite some time, in fact, before I decided to really try to think about what they might be. And I'm going to show you a movie now that we've made in my lab at Trinity, but this is essentially what I saw. This is many times speeded up. In fact, this movie was shot over a 24-hour period on a high-resolution microscope. And what we're seeing here are cells that this is a nice healthy cell, and you can see that these cells have embarked upon a process where they're kind of writhing in agony. They're doing something, clearly, something that's very different from the normal state. And in fact, what they're doing is they're entering this process of programmed cell death. And what you can see here is these cells are actually still alive by all criteria, except they've activated a set of enzymes inside that is actually dismantling the whole cellular architecture. And that is, in essence, what happens during programmed cell death. And then finally, at this very late stage, this cell now enters the process. And as I say, this movie is taken over a 24-hour period, so you'd never be able to observe this just by looking down the microscope. The steps would be too incremental for you to notice any change. But you would notice that at the end of the process, cells look like this, and clearly that's very different to normal-looking cells. So I was thinking about this for a while, and I realized that actually this was this process that had been very recently described, but really nobody knew about it. And this process of cell suicide or programmed cell death was considered to only take place under very special circumstances. And in fact, under the circumstances that I was investigating, it, by almost definition, it wasn't supposed to be occurring. But being young and naive and stubborn, I decided to pursue this. I was convinced that these cells were dying by programmed cell death, even though they shouldn't have been in this particular situation. Because at that time, programmed cell death was only known to occur, was only supposed to occur, at least that was the prevailing thought, in situations where cells were dying very naturally under normal physiological conditions. But here we had cancer therapy drugs that should be kind of causing this noxious type of cell murder, was actually causing a more programmed or natural form of cell death. Now, I mentioned necrosis earlier. I'll mention it again here, and I'll just show you a little movie again to contrast this with the type of cell death I've just shown you. Necrosis is generally thought to happen under non-physiological, pathological conditions, so in response to severe injuries, essentially, or noxious drugs and so on. And up to this point in time, this was the late 80s, early 90s, essentially most cancer therapeutic drugs were considered to kill cells via necrosis. And this is what it looks like. In fact, this movie is shot over a much shorter time frame, about an hour, in fact. So necrosis is very swift, very sudden compared to programmed cell death. And not a lot goes on. The cells, you can probably make out that cells are kind of bursting. And every cell in the dish bursts pretty much over the same time frame, within about an hour of application of, in this particular case, 10% alcohol. So in necrosis, you usually have large groups of cells all being killed, essentially, being murdered together in a group. It's very different from programmed cell death, where the cell initiates the process independently from its neighbors. This is another way of thinking about necrosis. These are some members of my lab when we were at a trip recently. So during necrosis, it's very swift. Everybody dies together, and there's not much you can do about it, really. So for those of you who remember what libraries are, nowadays we have everything accessible at our fingertips on the internet, and increasingly my students moan if I ask them to, in fact, take a trip to the printer downstairs, never mind the library. So in those days, when you wanted to know about something, you went to the library and you hit the books, and so I did that. And I wanted to find out as much as possible about this kind of weird mode of cell death that I had just been observing in my experiments. And I found, in fact, that there wasn't a lot really known at that stage. In fact, at that stage, you could read almost all of the literature in this area in the space of about a week. There was about 200 papers that had been published on this process over the previous 25 years. So this phenomenon had been recorded in a very kind of phenomenological way, and there was really very little known in molecular terms about this process of cell suicide or programmed cell death. So after satisfying myself that what I was observing was actually real and was interesting, I decided this was something I really wanted to work on, because practically nothing was known at that time about this process. And I immediately started to think about what genes and proteins could be involved in regulating this, and what kind of conditions could cause this, and would we be able to manipulate this type of cell death to, for example, cause a better outcome in disease. Because as I've mentioned, most diseases fundamentally are underpinned by either too much cell death or too little cell death. So there's a couple of points I want to touch on about the scientific process, and I think a lot of us, when we read about science, we think, as we heard by the president earlier, we think it's kind of dusty and boring and full of mathematics and boffins, eggheads in white coats, and people who spend a bit too much time indoors and don't have lives. Well, it turns out that science is actually a pretty special career. It's tremendously exciting, and when you realize that you're seeing something that possibly no other human has seen before, it's tremendously stimulating and tremendously addictive, and you want to have that feeling again and again. It's really remarkably inspirational, and actually a privilege to get to do things that possibly have not been done before. Now, we often think about discovering the universe as being, and obviously there's a lot of talk about a trip to Mars. At the moment, the universe is our unexplored frontier. Well, actually, there are vast tracts of unexplored frontier within our own bodies, things we just have no clue about. And these kinds of problems are still there as huge uncharted territories that still have to be explored. And as we develop new instruments to be able to look inside cells, we get to do this all of the time. Now, of course, sometimes you think you've made the discovery, but it turns out that you've just not read enough. And it does turn out sometimes that when you have what's called commonly the eureka moment, it's just a reflection of your level of ignorance. And in fact, I think it was John F. Kennedy said at the beginning of the space race that the greater our knowledge increases, the greater our ignorance unfolds. And it is very true that very often, until you've read a lot about a subject, you really don't know what's new and what's not new. So there is a danger sometimes when you have the eureka moment that in fact it's just a eureka moment for you. Everybody else knows that. But I'd pretty much established in my initial reading about this process that there was something interesting there to study. Now, Patrick Durier, who used to be in my lab, Patrick's from France. I just thought I'd show a couple of photos of Patrick illustrating the eureka moment. He was always very expressive. Sadly, this turned out to be a false alarm. So anyway, but that is a very special feeling, and it is one of the great attractions of doing research, that we get to feel sometimes that we are kind of sailing into completely uncharted waters, albeit with our microscopes or our DNA sequencers or our protein sequencers, but it's exciting nonetheless. So how does the cell know when to die, and how is this actually regulated in fine detail at the molecular level? That's the question that I've been pursuing. Now, people also ask me very frequently, members of my family, for example, have you had any breakthroughs lately? It's when you're having a chat, it's the common question that you get. And I always find this a very difficult question to answer, because it's very often in science very hard to know whether you've had a breakthrough, because sometimes that's clear, but in most cases that's not very clear until, in fact, sometimes many years after you've made the breakthrough, because especially in biology, nature is very illogical. This is something I found on the internet, but it's kind of a depiction of what I think the Americans call a Rube Goldberg machine, something designed in a very complicated, bizarre, indirect way to achieve something that may be quite simple. And in biology, in fact, many of our systems that operate in our cells are actually designed in that very illogical manner, because of course this has happened through Darwinian evolution. It hasn't been engineered, it hasn't been designed like an Apple computer or by some kind of engine. It is actually just evolved in a very random, indirect way. And so although there is a general conservation of certain principles from one organism to another, in fact, it turns out that each particular organism very often devises its own particular solution. So very often when you're investigating a problem, you don't have a template, you don't even have all the pieces of the jigsaw. You think you have certain bits of the jigsaw, and then after you've been putting together a particular corner of the picture, now you find another few pieces. And so the picture's always changing, and the number of pieces in the jigsaw are always increasing. So it's very often in biology, like a lot of the other sciences, it's very often a long road to fully understanding a process, and it can be difficult to know when you've made a breakthrough sometimes. Sometimes it's very clear, sometimes it's not so clear. And one of the reasons for this, as I mentioned, that cells are actually, contrary to the kind of simple depictions in the textbooks like this or this, cells are actually unimaginably complex, and we're realizing this more and more. And in fact, if this is a depiction of a cell, and this is a reasonably good depiction of a cell that you would see even in university textbooks, well then this is a pretty accurate map of Dublin in the same, drawing the same parallel. So that is as accurate a map of Dublin as that is an accurate map of the cell. This is actually a photograph taken with the Hubble telescope of a star cluster. And just in the way the Hubble telescope has transformed our ability, obviously, to see to the far reaches of the galaxies, super resolution microscopy and other methods have enabled us to see into cells. And this also looks like a picture of a star cluster, but in fact this is just 1% of the area in one cell, looking at some of the proteins, and the yellow and red are just two particular type of proteins, and each of those dots would represent hundreds of individual protein molecules working in little clusters. So this shows you and I'm going to just zoom out here on this actually shows you the problem that we're facing with in order to just understand one cell. And that is not by any means lighting up all of the proteins that are present in that one cell. So I've mentioned the word protein a number of times. I think many of you here know what proteins are. Essentially, they're the enzymes that do the main heavy lifting in the cell. So cells are comprised of millions of individual proteins, or not millions of individual proteins, but millions of copies of proteins. There are about 10,000 individual proteins in a given cell, but up to 1 million copies of each one. And this is a molecular snapshot of some of the proteins in a cell. There are methods we can use now to resolve these proteins into their individual molecules. And that's only a snapshot of about 10% of the proteins within one cell. So every single dot, no matter how faint, on that snapshot is actually an individual unique protein. So this is kind of what we're dealing with. We're dealing with the situation whereby a cell is a bit like a city. Each individual cell is like a city. It has its power stations, its mitochondria. It has its press offices, like the Irish Times. Its press office in this case actually makes communication molecules called cytokines and growth factors that releases and disseminates information to other cells. Waste disposal used to be the LIFI. I guess we have a more sophisticated way of doing this now. Transport hubs, microtubules, actin microfilaments, and so on. And it literally is like a city. It has that complexity. And if a cell is like a city, then the proteins are the individual workers within the city. So proteins are like people. They actually do the job of work within the cells. And they're actually subdivided into businesses, into little units of activity. But the problem is we have no idea who does what in the cell. We have some idea, of course, but there are many, many different individual businesses in cells, and we essentially go about trying to discover what everybody does by effectively reaching in and pulling out some of those individual workers and trying to figure out who's relevant and who's not. So I think this is a good analogy here. This is a restaurant, and imagine this is a little compartment in the cell. Well, the only really relevant people in terms of actually producing the food in the restaurant are obviously the waiters, the chefs, and obviously the people who wait on the tables, bring the food to the tables. The customers can change every night. And with protein communities in the cell, with these little businesses, there are a lot of red herrings. There are proteins that are an integral part of complexes, and there are proteins that are not. So that's one of the reasons why it gets complicated to study a process like programmed cell death, where you're trying to find out which proteins are actually relevant to the process and which proteins are kind of only guests or customers. And as I mentioned now a couple of times, because there are on average about 10,000 different proteins in even one cell, and many, many copies of each individual protein, finding out who does what and when and how can get to be very complicated. So frequently it feels like this is what we're doing. In the way that we resolve things sometimes, it's just the methods we have available to us right now are completely inadequate. And also sometimes we're looking at things totally the wrong way around. And I just want to show you a rather trivial example of this. So I was at a scientific meeting about 1.5 years ago in Cape Town, South Africa, and a friend of mine who's a scientist brought us on a bit of a tour of the Cape because he used to live there. And we came to this very nice bay, and I wanted to get a couple of photographs, and as we all did, in fact. So I took a photograph of a colleague, friend of mine, Guy Salveson, and he's a really excellent scientist. In fact, he discovered one of the key families of enzymes called caspases that actually regulate programmed cell death. So I took a photograph of Guy at this very attractive location, and then I handed him my iPhone and I asked him to take some photos of me. And this is what I got back. And my personal favorite. So even scientists can make mistakes. And in fact, we make mistakes a lot. And sometimes we come up with hypotheses and conjectures that are just wrong. And science, like any other human endeavor, actually, is a process that's imperfect, and it corrects itself over the years. And in fact, this is another reason why progress can be slow. Scientists effectively police the gates of the scientific literature, and they do so with great brutality at times. And this is a cartoon I found, and it's quite a well-known one. There's a process in science called peer review, where when you write up your discovery, you submit it to a scientific journal, and the scientific journal then sends it out to, anonymously, 3 or 4 scientists working in your field, and then they proceed to beat the living daylights out of your hypothesis and give you another year's work, or in fact tell you that this should never see the light of day, and while we're at it, here's another rejection slip for your next paper you might be thinking of submitting to this journal. So the process of actually making and actually also publishing, disseminating discoveries in science can be quite slow and quite painful. So back to programmed cell death. Over the last 20 years, we've learned a lot about this process. We know that it happens everywhere. As I've mentioned, it's used very extensively in the body to get rid of cells in all sorts of situations where cells aren't doing their job properly, or just because the cells are only needed for a small period of time. And in fact, during development, when we're developing as embryos in the womb, there are lots and lots of situations where cells need to die just to build something. This is like a process of sculpture, just the way a sculptor starts with a crude piece of marble or block of wood in order to carve out a very nice finished sculpture. Nature does the same thing. It overproduces cells, and then it kills off cells at places where it doesn't need them. Programmed cell death is also used, as I've mentioned earlier, to cull the weak, the sick, the injured, the abnormal, the weird, cells that are just not functioning properly. So it maintains the vitality of tissues. It's used, as I say, to replenish our tissues on an ongoing basis. Now, here's a couple of examples. This is the developing mouse embryo, and initially what happens when you're developing a limb is that a crude limb paddle is made, and this is the limb bud here, both for the legs and the, sorry, this is jumping ahead. So that crude limb paddle is then sculpted. Where those arrows are, the cells at those precise locations during development are progressively killed off. So initially we have webbed hands and feet, and then in the final form, you develop the digits by carefully triggering programmed cell death in the cells that need to die. Now, this is actually from an example of a small eelworm, the nematode, Caenorhabdus elegans, where 10% of the cells in this animal die during development. And it's been a very useful tool for doing genetics to dissect the genes that actually regulate this process. So we're seeing the development of this embryo here over a period of a number of hours, and at very precise times during this developmental process, 131 cells are actually killed, and they always die at the same time at the same place during development, which is remarkable. This is actually an inbuilt program to produce cells whose only fate is actually for a couple of hours during development, and they're needed in order to build certain structures, and then they're no longer needed. So which genes, proteins control the process of programmed cell death in humans? As I say, that's been a fairly long journey to figure that out, and that's really started, as I mentioned, during my PhD. We started to work on this, and this was before this process was being studied by very many labs. This is me around that. So this is my Joy Division phase, if anybody remembers that band. But basically, we knew at that point that certain triggers initiate the process, but we have absolutely no idea what went on in the cell in order to execute the process finally. I then went to University College London to study with this man here, Professor Ivan Reut, who was a very famous immunologist, and in fact was very well known to me. I used his textbook during my undergraduate and postgraduate studies. And so he put me to work on a problem at that time. I actually thought I was getting away from cell death at that time. It's not that I'd had enough of it, but I was interested in learning about the immune system. And at that time, HIV was really the major infectious disease problem in the early 90s. And in that particular condition, essentially this virus progressively kills off a particular type of cell in your immune system called a T cell, and that leads to AIDS. And what's actually remarkable in the last 20 years is that disease has been actually transformed through research from something that was invariably fatal within 3 or 4 years of diagnosis to now a chronic long-term illness that is treated by protease inhibitors and other antiretrovirals. But at the time, there was no therapies whatsoever available. And so one of my tasks was to figure out what was happening when the virus entered the cell. And what I found out very quickly was, again, this process of programmed cell death starting out from under the microscope very soon after cells were infected. And that was, again, a surprise at that time that, in fact, the HIV virus actually switches on the programmed cell death machinery. And if that fails, there are specialized types of cells that, in fact, we've also been studying over the last number of years. And these are called natural killer cells. They're cells of our immune system. And they're there to effectively kill or execute a cell that may be infected with a virus. And I have a movie of this. So if a cell is infected with a virus, either it should kill itself or it will be killed by this special assassin cell, this executioner that's present in the body that goes around looking for virally infected cells. And we know now very precisely how it does this. It actually delivers enzymes into the virus-infected cell, which causes it to die. And there's a movie, again, shot in my lab a few years ago, and you're going to see this cell here is going to be attacked by this cell here. At least it was this morning when I played this movie. So hopefully that will also happen again. And something has gone wrong there. Oops. Let's try that again. So the cell there that the arrowhead is pointing on should be attacked. And what's going to happen there is this natural killer cell is going to deliver essentially an enzyme that's going to switch on the programmed cell death machinery of the target cell. So it's now actually delivering, it's degranulating, it's undergoing this process of agony, and then the cell that it's attached to will now actually die. And you'll see that one or two of these other natural killer cells in the vicinity go a little bit crazy. They get very excited by what's going on there. So this is moving all around the place. Anyway, this is a cell that's actually now been effectively executed. And that's exactly what you'd like to happen when a cell in your body is infected with a virus. Either it kills itself, or if it can't do that, because a lot of the viruses actually try to prevent that from happening, then it's executed by the special assassin cell. So during that period of time, the kind of molecular machinery for cell death started to emerge, and at that point, these enzymes called caspases that chop up other proteins had emerged as the key regulators. We still didn't know very much about it at that time. And then I headed off to California, where at that time, this person here, Doug Green, was working on this process. He was one of the noted authorities on programs like that at the time. So as a young postdoc, I headed off to University of California, San Diego, to start working and really trying to learn molecular biology, because I knew that was a skill that was required to really start to tear apart the machinery of this process in any detail. And this was kind of the Klondike gold rush phase of cell death research, because a lot of different scientific teams had moved in on this problem, and there was really a lot of frantic competition at the time to I guess, win the race to discover a lot of the different things. One of the things that we did when I was in Doug's lab was discover a molecular probe that is still used today to identify cells that are dying by programmed cell death. And a lot of different companies sell this particular molecule in XM5 that's used as a diagnostic to detect dying cells. And this is actually a movie that was made in Doug's lab at that time. Again, this is another view of cells dying by programmed cell death. The cell is going to go kind of purple-red as this probe that we developed binds to the cell. And because of the nice kind of change in coloration there, Doug calls that movie Peace, Love and Understanding. He's Californian. A remarkable thing that happens when cells actually die by this process is that they're eaten by their neighbors. And this molecular marker I told you about a moment ago is a change that takes place on the cell membrane that actually gives permission to the neighbor to eat it. Now, again, this is breaking a fundamental rule in cellular society. Just like in human society, we never give permission to our neighbors to eat each other or to eat ourselves. And cells don't normally do that in the body, but in this particular case, this acts as a kind of a efficient disposal mechanism. When a cell is dying in this kind of regulated way, it gives permission to neighboring cells to eat it. They swallow it up before it can burst open and cause any damage. And it's a very nice, eco-friendly recycling process. So the pathway was becoming more complicated now. We discovered, along with many other groups, quite a few additional molecules that are involved in the regulation of this process. And I'm clearly not going to bore you by going through the intricacies of it, but when people ask me sometimes what happens in programmed cell death to kill the cell, I like to show them this photograph, which I can't remember who sent me this a number of years ago, but apparently this person was painting his front room and went out to take a phone call and left a can of paint with his children there for about 10 minutes. And came back in, and this is what they had helpfully done. So this is kind of what happens when caspases, the enzymes that regulate programmed cell death, they're activated in the cell. So they kind of set up this explosion, and they kind of progressively wreck the cellular architecture. And that's actually a subject that I've been working on for about the last 8 or 9 years, understanding how caspases coordinate all the events that take place in apoptosis, and it gets very complicated. So I moved back to Ireland, and I've been in Trinity now for about 13, 14 years, and I have a great group of people working in my lab. Of course, people come and go over the years, and death has, of course, got a lot more complicated. We know a lot more about this process now, and there are many, many molecules involved, and it all gets very, very complicated, far more complicated than I could have imagined when I first started to work on this process about 20 years ago. So why do we care about this? Well, I think it's important that we understand biological processes for their sake, because this is This is how we are. This is how we work. It's important to understand that. But it's also important to understand it because clearly something that's so fundamental as cell death clearly goes wrong. And when it does, it causes disease. There's a nice illustration of this. This is actually from a mouse. And these are normal lymph nodes, these little white dots here from a normal healthy mouse. And when you mutate just one gene, that is involved in the regulation of programmed cell death, in the bottom dish here we have 4 lymph nodes from a mouse that's mutant for one single programmed cell death gene. And what happens there is those mice accumulate many extra cells because the cells don't die at the normal rate. And so very quickly those mice accumulate huge amounts of extra cells and they develop an aggressive autoimmune condition similar to lupus. Of course, the same happens in cancer. We now know that many cancers arise not because you have extra cell division, but because in fact you have too little cell death. And there's the other side of the coin. When you have an accelerated rate of cell death, you can get various diseases such as neurodegenerative diseases. In Parkinson's disease and Alzheimer's disease, you have an accelerated of neurons in certain areas of the brain. So if you disturb this balance, this equilibrium, and there are mutations in genes that are involved in some of these cases. So Parkin itself, for example, is mutated in Parkinson's patients. There are mutations and other reasons why this equilibrium is disturbed, and that can result in disease. And that's one of the reasons, of course, why we want to identify the molecules involved. And in fact, there's been tremendous progress in understanding how we can manipulate cell death in disease settings. And of course, one of the big reasons is because in cancer, in fact, the main treatment in cancer is either to take out the tumor or kill the tumor without killing healthy cells. And in order to kill those cells, you have to know how to effectively turn on the programmed cell death machinery. And we know how to do that in much more exquisite detail with much more precision than we knew how to do that 20 years ago. And there's a whole suite of molecules now, some of them are pictured here, where we really understand how they work in very fine detail indeed. And there are very sophisticated therapies that are actually now being developed as a consequence of the accumulation of this knowledge. So I'm near the end. I just want to finish with a few remarks for if there's anyone in the audience who's a budding scientist who's wondering, should they consider research as a career? Why do we do it? Why should we do it? I think those reasons are obvious. I think these days there's a temptation that everything should be justified in economic or job creation terms. I think that's a mistake. Clearly science should be done because knowing more about ourselves I think has served us very well. Our natural curiosity separates us from most of the other animals on this planet. Look at the surroundings that we've managed to create for ourselves. Look at the devices, look at the knowledge, look at the kind of medical treatments that are available to us. And we're the only animal on this planet that is able to control our environments with such remarkable precision. So we need to do research to advance the human condition, to advance the body of human knowledge. We need to understand our world ourselves because we're there. It's the way we can cure illness and the disease. And from a personal perspective, to pursue research as a career, it's a fascinating career. It's no 2 days are the same. It provides an ongoing challenge, and it's something that if you fall in love with, you'll essentially never go to work again. So I'll finish up on saying that, as I've said earlier, cells are tremendously complex. There's still so much that we don't know about them. We're probably 20 to 30 years away from a detailed understanding. And so those of you who are thinking of going to research, I would certainly encourage you to do that. It's a very rewarding career. And it remains for me to thank the remarkable people, incredibly dedicated people who've worked in my lab over the last 12 to 14 years or so. Of course, these are the people who actually do the work. I write the papers and I get to give the talks and fly to nice places. These are the guys who work 12-hour days and very often with very little pay. And there have been a number of generations of people in my lab over the last 14 years or thereabouts. I'm just going to put up a few slides. If some of you are here, you'll recognize yourself. I'd also like to thank my colleagues within the Smurfit Institute of Genetics at Trinity. especially Professor Tony Kavanagh, who I see is here, who nominated me for this award. Thank you very much, Tony. I appreciate your support. And also thank you to people from Science Foundation Ireland who funded our work for the last 12 years or so. Thank you again to our sponsors, the Royal Dublin Society and the Irish Times. Please continue to support science. It seems that at times that's not done enough. Science seems to be much less important than sport. Sometimes it occurs to me, but anyway, I think scientific endeavour is a very worthy thing to support. And finally, my best experiment ever, they're sitting right there, and I'd like to also thank them for just being them. And also, thank you, Mum. My mother has always been a constant source of support, inspiration. I still call her when I'm moaning about things, and she still listens. So thanks again, Mum. And thank you again for listening. And it's, again, a great honour to receive this award. Thank you.